Entrain is a computational tool for single-cell RNA data that elucidates the extracellular drivers that are responsible for differentiation. It does this by merging trajectory analysis with ligand receptor tools. So, in multicellular organisms, cells adopt different states of differentiation, which allows them to perform their specialized tasks. This requires both multiple cell fate decisions, as well as, importantly, the ability of cells to communicate and regulate each other to perform their tasks. So currently, cell fate decisions are studied by profiling the gene expression of single cells and arranging them into a developmental order called pseudotime. And there's currently more than 70 published methods for this called trajectory inference. And this reflects their popularity as well as a need for specialized methods. Of course, specialized projects, require specific tools. And so an alternative approach, RNA velocity, uses the relative abundance of unspliced and spliced mRNA to calculate the time derivative of RNA expression. And in principle, this allows one to predict future cell fate. Now, in parallel to all of this is development of a completely separate set of tools called ligand receptor analysis. And this has been largely developed independently of trajectories. This approach has made it possible to decipher cell-cell communication that may also regulate cell fate transitions. And so methods such as this, such as NicheNet, take advantage of a large body of prior knowledge about gene regulation, intracellular regulatory networks, and so on, to identify more active ligands and their flow-on perturbations in gene expression. Therefore, these tools actually have the potential to complement trajectory inference and RNA velocity by providing an orthogonal set of data. So in this project called Entrain, we have integrated these two data sources. So by merging these information sources, this tool is able to identify the extracellular drivers of cell fate transitions and also identify the cells responsible for regulating differentiation. The algorithm consists of three main steps. So first, we construct developmental trajectories using established methods of trajectories, such as monocle or RNA velocity. From these methods, we select genes that are most informative of the dynamics, and we call them training genes. And so in the case of pseudotime, training genes are selected on the basis of their covariance with pseudotime. And in the case of RNA velocity, these genes are selected on the basis of their velocity likelihoods or confidence, which is recovered from the function recover dynamics in SC-Velo. And so in parallel to all of this, we also identify ligand receptor pairs and the downstream regulatory networks using NicheNet. And in the last step, we merge these outputs using a random forest regression model, using the training genes as an independent variable and the NicheNet prediction as the explanatory variable. And lastly, we visualize the results. So we can demonstrate the utility of this on two datasets. The first one is here. This is a hematopoietic bone marrow dataset from the mouse. And we have corresponding lineages of developing osteoblasts and pre-B cells and so on. Firstly, running on the Entrain on the osteoblast population results in these key ligands responsible for that differentiation, such as TGF-beta and DKK1, and these are actually well known in literature to be driving osteoblast maturation. Similarly, for the B cells, we recover IL-7 and B-cell activating factor, otherwise known as TNF-SF13B. And these are also very well supported in the literature. And interestingly, we also recover laminin-2, which has not yet been supported in literature. However, we found that its receptor integrin beta is actually necessary for maintenance of the B-cell compartment. So in comparison, existing tools such as NicheNet and CellCore, they're not actually designed for trajectories. And so therefore, they actually have quite different results, suggesting that we can actually improve our results by using trajectory informative genes like we have done here, instead of the more traditional differential expression that's normally used. We can also run Entrain on data sets more compatible with RNA velocity. And so here we have run SC-Velo on an embryonic neurogenesis dataset of the developing mouse embryo at gestation day 10. We've clustered the velocities into 10 main groups of velocity vectors shown in these colors here. And these roughly correspond to the major cell state transitions, for example, neuroblasts to neurons in the bottom right. Using SCVelo, we have recovered the genes that exhibit the most confident or the most rapid velocities, and inputting these into Entrain produced the ligands shown in the boxes. We can see that 5 of the clusters have positive variance explained, which is a measure of the model's fit quality. And so the positive ones imply that they have some component that is environment dependent, while the negative ones imply are not well explained by the environment and possibly are controlled by cell intrinsic mechanisms like transcription factors. So cross-checking the ligands of the 5 positive clusters shows that 12 out of 15 are actually involved in neurogenesis in sort of the literature. And this is a significant improvement in comparison to other tools because they cannot actually incorporate RNA velocity. Again, they only use differential expression. So we can see that incorporating velocity has also improved the identification of extracellular regulators. So in conclusion, we have developed this package called Entrain, which merges 2 previously independent lines of single cell analysis, trajectories and ligand receptor analysis. The result of this integration is a much improved identification of the ligands and the extracellular regulators that are responsible for driving different cell fate decisions.